Uh, it's indeed it's a great pleasure and honor to me to be here. I'm also very thankful to the organizer of this conference, international conference on advanced material for sustainable innovation. So I'm here basically we have worked on various sensing platform, particularly on optical fiber based sensors that we have simulated, manufactured, developed, tested. But today I'm sharing some of our work on the optical fiber biosensors, like what is the current is going on and what we can do in the future and how they can basically make the innovation for sustainable development. So basically I'm belonging to a great organization that is called the CSIR, Council of Scientific and Industrial Research. As far as the students is concerned, they know CSIR as a body who is providing the fellowship for their research, their PhD, and of course some of the grants to the faculty members in the across the country of various institutions using different funding modalities. So actually CSIR is having almost 39 laboratories across the India, and they are working in various all the areas, whether you talk of material science, aerospace, biology, medicine, plant, instrumentation, semiconductor, you name any area. So the CSIR labs are there, and they are making their footprint, and they have made lot of innovation, and they have provided lot of technology to the country. All of you know, like during the election, there is a ink, indelible ink, and that ink is developed by the CSIR, one of our laboratory. Even the this milk powder was when there is a scarcity of the milk in the country, the CSIR, our one of the sister laboratories, they have developed the milk powder at that time, right? So we are working in many fields like healthcare, agro, food, chemicals, ecology, environment, earth sciences, aerospace, mining, material, housing, construction, everywhere, right? So about the CSIR, like about our own laboratories, so its origin is like from by the Planning Commission, established in 1959, and this is the picture of our first Honorable Prime Minister Jawaharlal Nehru, Pandit Jawaharlal Nehru. So our laboratory is basically working in certain areas like agriculture, healthcare and public safety, energy monitoring management, of course optics and photonics, strategic instrumentation. So actually optics and photonics is my domain, and basically some of the unique strength of my laboratory is like human resource. We are having the CSIR, so which is providing the PhD for the science and engineering students. We have a PRISM scheme, so where the some of the startups or innovators, they can submit the proposal. If the proposal is very innovative and it's doable, so they can get the funding up to 20 lakhs, right? So that is also one of the very unique scheme in the country, which is our laboratory is also one of the nodal center, and it is from the DSIR, Department of Scientific and Industrial Research. And also if your proposal or maybe if it is then you have demonstrated a prototype, then further you can get the funding of 50 lakhs for the startup. Then we have a skill development. So through the CSIR and skill India Skill Innovation, we have one of the unique Indo-SWIFT Training Center, where we are providing the diploma, so 4-year diploma to the students. So we have a very good capabilities. so which can cater the MSMEs. So this is my like I have enlisted my talk, like I have just give an introduction, and then what are the fiber optic sensor. I know many of the students, they are from the IoT background, like they are doing the IoT, BTech in IoT. So sensing or sensors are very important for the IoT, Internet of Things, because without sensing integration and their basically emulation, like you can't do the IoT, whether it is agriculture field or any public safety or anywhere. So sensors are the integral part of our IoT platforms. So and then of course, because I am talking on biosensors, right? So I have to just introduce how to make the optical fiber as a biosensor, and then of course I have covered some of the things like these are the different kind of fiber platforms, like they are called the different names, like multi-mode fiber brake grating, and then I have given some case studies like we have used for this sensing platform. For example, for the bacteria sensing, and then biomarker like triglyceride, we have also detected the triglyceride. Then we have, because this conference is related to material, of course this talk is slightly different, because I am not talking about the material like how our previous speakers, they have spoken about the material of the magnesium to many good novel material. So of course, but we have used these materials to exploit their good features to get the good sensing modalities. Then of course, because I am talking on the optical fiber, but the future is basically on chip photonics biosensor. So I have also included some of the on chip sensing platforms and how they can be simulated and how they can be fabricated. So and of course initiative also have covered the initiative towards the on chip photonics, what we are doing in the CSIO, and then conclusion. So as you can see, the sensors are the device like which can convert one of the physical parameter to the magnitude of a second different parameter, which can be measured more conveniently and more accurately. And as we know, like these are the different sensors people are using, and as we know, during COVID-19, in every household the people are using the pulse oximeter to measure their oxygen content, right? And that at that time it became a very important device for everybody, right? And then in the older time, in the mining, people are using this bird just to detect the methane outburst in the mine industry. Thermometer we are using, like glucometer everybody using to detect their diabetic condition. And this one of the COVID-19 kit was developed by our sister lab in the New Delhi IGIB. So it's a paper strip, so that can detect the COVID. You know, like importance of the sensor research, because we know like there are lot of pollution through the industry. Then of course, you know, there are lot of pesticide people are using in the agriculture. So you should know like what is the level or what is the contamination of the pesticides, whether it is a permissible level or not. So you know, they all are posing a lot of threats to the human life. And of course the industrial based, because the industries are due to their you know, they are due to their profit and all, they are not looking how they are polluting the environment, our water bodies, and also you should also detect the water contaminations. Then bacterial infection, people are also on a present day talking on the food safety. So there are a lot of people are using the sensing modalities to detect the food contamination. And of course there is a lot of market for the sensor research. So and it is keep on growing. For the students, like these are the some of the parameters, like how you can define like a good sensor is, because for the researcher also These are the some of the parameters like sensitivity, figure of merits, quality factors, signal to noise ratio, limit of detection. So how like people are researchers, they are using in the all their studies and also in their research papers. So if you don't have these parameters in your research papers, so definitely a reviewer will ask you to do all these things, so then only that paper can be considered it's a good quality. Okay, so these are the some of the process for how you can classify a biosensor. So a biosensor is basically it consists of bioreceptors, which is called the biorecognition molecules, right? It can be antibody, it can be aptamer, it can be biophos if it is a bacteria, it can be many kind of bioreceptors are there. And then there is a transducers. Transducer can be the amperometric, optical, then fluorescence, many kind of transducers are there. So then by integrating both of them, they can become the biosensor. Like you have you wanted to detect some bacteria, you need to have some antibody, the bacteria will come, and then that particular antibody will interact only with that particular bacteria, and then that can change the signal, and that the signal, the change in the signal which you are wanted to detect, the signal is basically the your transducers, like whether you are detecting in terms of the current or in terms of the fluorescence quenching or fluorescence enhancement, or maybe change in the wavelengths, or maybe the change of different kind of signals. So that can be the become the complete biosensor. Okay, so these are the I have also covered, like because I'm talking about the optical fiber. So these are the some of the research groups in India, like IIC Bangalore, IIT Patna, IIT Delhi, and many others, like many other labs also, they are working on the optical fiber based biosensors, right? So they have detected like E. coli bacteria, then they have also used this grating for the this sensor of sensing of the temperature and strain. Then they have also used SPR sensor for the cancer cell detection. So these are the some of the groups, those who are really working in these areas, right? So then I'll come to the optical fiber sensors. So optical fiber sensor is a sensor that measure a physical quantity based on the modulation of its intensity or spectrum, phase, or maybe the polarization of the light traveling through the optical fiber. Like any change, like let's say if I have optical fiber, so you all know the optical fiber is used for the optical communication. So like we are enjoying all this kind of video, and then very high bandwidth in the mobiles. So that backbone is basically optical fiber, which is carrying the terabytes of signals, right? So optical fiber is very small in dimension, like 150 micron diameter, and the 250 is total diameter including its outer jacket. But when we are using, so its dimension is as you can imagine equivalent to your human hair, right? But it is carrying a lot of signals, right? So similarly, like when there is a change in the intensity, like the light traveling through the optical fiber, so the people are, you can make, they are making the optical fiber itself as a sensors. Like if you send some light and you detecting at the end, and if there is change in the signal, so it can make a intensity based sensor. Like people are using intensity based, like pressure sensor or maybe the temperature sensors. So similarly the phase are like for the interferometer, polarization based and wavelength based. But the advantage, if you talk of advantages, so optical fiber they have lot of advantages as compared to the conventional sensors, whether they are the strain gauge electrical sensor. So the most important advantage is optical fiber is made of the silica. So that is basically kind of material which do not have any impact of the RFI EMI radiation. So, even if the, let us say if you have some environment where you have some electromagnetic interferences, right, or maybe the some of the environment like where the electromagnetic pulses like nuclear radiation environment. So, where the conventional sensors they are not working, right. So, the fiber optics, because they are insulator basically, right. So, you can use those kind of sensors, like you have sensor. So, and another advantage is the sensor and the channel is the same, right. The sensor itself is a channel. So, the fiber is a channel also and it is a sensor also. So, you do not need to have a separate kind of line to send the signal, right. So, and then it has a multifunctional, like you can make in one signal fiber different sensing modalities. So, you can one sensor can sense the temperature, one can sense the strain, one can the pressure, one can the vibration. So, like in one single channel a person can use the multifunctional capabilities. Remote accessible. So, the optical fiber that is having the 0.2 dB per kilometer losses. So, you can monitor from the kilometers of distances, right. So, like you can have your station monitoring station somewhere else and the site somewhere else, then also you can use this kind of sensing platform. Okay, so, these are the, I have also just have some introduction of the what kind of sensing platforms. So, and that is just because the students they might not knowing. So, these are the fiber Bragg grating. So, Bragg, you know, like the Bragg, the name of the scientist, right, who has given the Bragg principle, right. So, about the Bragg diffraction, right. So, similarly the similar principle have been used in fiber Bragg grating, like fiber is the optical fiber, Bragg is the, because it is working on the Bragg condition, and then grating is the periodic structure. So, because these are the periodic structure, right, and that we have created using the UV light, because and this structure was created in the core of the optical fiber, and these structures of the nanometer dimension, like they are the 500 nanometer, and that we have created using the interference UV lithography, right. And then this structure basically if you send let us say a broadband pulse, right. So, it will not allow to pass all the wavelengths, one wavelength is reflected back for which this structure is created and all other wavelength they pass through. So, they are also working as a wavelength filter. So, this kind of device used in the optical communication also for the eye drop multiplexing, right. If you are going your signal or your one channel is going from Delhi to Chandigarh and you want it to drop in somewhere, so that this can be used, so that the channel will be dropped. If you want to add at certain location, that can be used for the adding the channel. So, that is the communication perspective, but for a sensing perspective, basically if I use this device as a strain sensor, so I can just surface mount is or maybe embed in the some structure. So, if the structure is experiencing let us say some beam is there, like for the structural health monitoring in bridge or in a building. So, if the bridge is experiencing some stress or some bending, so there is a change in the its periodicity, and then there is the shift in the wavelength, and then one can detect the something is happening in the structure at that particular location. Another kind of sensor is basically long period grating. So, why that is called the short period grating? Because that periodicity is of the nanometer. Here the long period grating, the periodicity of the order of the micrometer, around 100 micrometer to the around 600 micrometer. So, here the thing is like those sensors which I have discussed, they are the reflection based sensor, right. So, you have sent some light and then one of the wavelength is basically some particular color, right. If you have sent let us say 10 colors, so then one of the color is reflecting back, right. So, these sensors, because they are working in the reflection mode, and these sensors, because here the thing is like because of this periodicity of the longer period. So, the core mode power is coupling to the cladding mode, and then you have evanescent field is available on the surface of the optical fiber, and if you wanted to detect some of the biomolecules, so you just modify this surface. If I think this is not coming, mouse is also not coming, right? No problem. Okay, the pointer is all not coming because pointer is all this. No, it's not working because it's not a screen. This is TV, so it's absorbing light. It is absorbing light basically, right? Okay, so no problem. So this part is basically we can manipulate using the bioreceptor, and then you can apply any kind of analytes, and then there is a change in the wavelength. So that basically become the sensor. Another important sensor is surface plasmon resonance, right? So where the optical fiber surface is coated with the nano layer of metals. Metal can be the gold, silver, right, or maybe ITO, right? So depending upon that at what wavelength range you wanted to operate. So people are coating generally gold, right, using the sputtering method. It can be the thermal also, or it can be the magnetron sputtering. We are also having the magnetron sputtering in our institute, and it is basically customized. So because the optical fiber is cylindrical structure, it's not the flat structure. So we have customized in that way, so that there should be a uniform coating on all the surface of the optical fiber, right? So the thing is, like, once you coated the any gold, any material like gold or silver, and when your EM wave, electromagnetic wave, is interacted with the metal layer, so the free electrons, they will be basically start oscillating, right? So because of the oscillation of the electrons, they generate the surface plasmon wave, right? And the surface plasmon waves, it is basically die out in the this one, and then you can get in the air, and you can get this kind of resonance wavelength at the output, right? So these are the output spectrum, right? Then important thing is, okay, now you have done everything. Okay, you have coated your surface of the fiber with the antibody and with the your bioreceptors. But how do you know, like, whether really there is a uniform coating, it is done or not done? So then there is some of the characterization techniques. So we have used the confocal microscopic just to analyze the protein attachment, like whether really the protein attached or not on the optical fiber surface. So you can see, because of the confocal microscopy, that the protein is interacting with the laser light, and then it is fluorescence, this green fluorescence. So then that is the basically confirmation that the protein attached on the optical fiber surface. And also people are using the Raman spectra for the antibody coated optical fiber, so that Raman is also giving the peaks for that particularly antibody. So that is the basically for the confirmation purpose. Now just I'll take you through some of the our studies what we have done. So using this kind of optical fiber, so like we have used first fiber break grating, okay, for the Salmonella typhimurium. It is one of the kind of bacteria, right? So it's a very dangerous bacteria. So that we have just for our just to test our platform, whether how much the sensitivity is there, how it is, whether really it is the sensor is working for this kind of detection of the bacteria or not. So what we have done, these are the some of the like design recipe. So where you can see, like, we have taken the optical fiber, and then we have fabricated the sensor also, because we have the facility to fabricate the sensors. So we have fabricated using UV light. So the sensor is sensor was created in the optical fiber core, and then the thing is, like, we have used the basically that is called the covalent bonding, right? Directly it is not attached on the optical fiber surface, right? You can just deep, so it bond. So we have to use some of the chemistry, right? So that is called the covalent bonding. That is also called the EDC NHS chemistry. right. So, the chemistry people, they know very well, because I am not from the I am not a chemist. So, we have our collaboration with the chemistry department. So, they have used this chemistry, covalent bonding. So, basically they have first used the piranha solution, that is the H2SO4. So, that's to generate the hydroxyl ion on the optical fiber surface. Then we have used APTES, so that it can generate the NH2 group, amine group. And because amine group is having a very good affinity, so with the this carboxyl group of the your antibody, so that is how that antibody is attached with the CO bond of the carboxyl group. And then we have a bacteria culture, like the students have performed. So, and those bacteria, they are the non-pathogenic, they are not, because to handle and just for the safety purpose, we have one of our laboratory in the Chandigarh, that is called the Institution of the Microbiology lab. So, they have basically cultured and they have given these bacteria, but we have ensured that they should not be pathogenic, otherwise they can create problem to the human being. So, we have used the non-pathogenic bacteria just for the this testing of the sensing platform, and then we have detected the this bacteria. So, you can see like these are the some of the tests we have done after, because we have reduced the diameter of the optical fiber. So, we have etched them using the hydrofluoric acid, and then we have used the same images, right, like to just to see that their platform their surface profile, right. And then you can see here the optical fiber, the portion, half of the optical fiber was etched. So, these are the some of the, you can see the result, because all the optical fiber based sensor, they are the refractive index based basically, right. They are sensing the change in the refractive index, that is the very sure, right. So, they are all refractive index based. So, you can see like we have first basically here you can see these are the RI solution we have prepared, and then we just tested like how much the change in the wavelength. So, just to calculate the sensitivity, right. And then because these sensors, they are also equally sensitive for the temperature, like if you have sensor and then you are want to detect some of the biomolecules. So, you do not know whether the shift is because of the temperature or because of the some molecule is there. So, that we have also tested for the temperature. So, what we have done, we have made in such a way. So, the half of the sensor where we are not applying any solution, so that is sensitive only for the temperature. The other part is sensitive for both temperature and the bioactivities. So, the amount of shift what we are getting from the this portion, that can be subtracted, and that the sensor can be make the temperature insensitive, right. So, that also we have done. So, these are the like you can see like we have detected very small quantity. The CFUs, they are basically classifying the bacteria colony forming unit, because the bacteria, they are forming the colonies, right. So, that is called the CFUs. So, then we have detected the 10 CFU, 20 CFU, 30 CFU, it is a very small quantity, right. And then you can see here these are this portion is enlarged there. So, then we are getting very good shift in wavelength, and here there is no shift because of the portion which is not etched. So, then there is no bioactivities. So, these are, and then also the important thing is, so you do not need only have to do the sensitivity study, but you have also do their selectivity and specificity studies, right. So, then in the last graph you can see we have also, because we are using the antibody of that particular bacteria, and then we have applied the different bacteria also, like B. serosus, and then E. coli, A. serosus, A. serosus. So, then they do not have much shift in wavelength if we are putting these bacteria. So, then these antibodies and these sensors, they are also highly selective for that particular bacteria, right. So, we have then come up with the sensitivity around 15.7 nanometer per RIU change, and then LOD is the 7 CFU per ml. So, basically the sensitivity if you are calculating, it is a very absolute quantity, but LOD is a basically statistical quantity. So, it is calculated from the standard deviation and also. So, this is like it is a standard deviation. So, it is a statistical quantity, right. So, that one must keep in mind, like what you are presenting, right. Then another sensing platform, as I have demonstrated the long period grating. So, where the long period grating also we have tested. So, long period grating platform we have done one of our collaborator in South Delhi. So, Professor Inakshi Sharma. So, with them we have done a study on the triglyceride detection, right, and this study was very also appeared in the one of the very reputed journal, Biosensor and Bioelectronics. So, here same, the chemistry is same as I have explained, that EDC NHS chemistry, only the sensing platform is different, okay. And also we have shown, sorry, also we have shown like when you have prepared your and we have used the lipase enzyme for the detection of the your triglyceride, right. So, you know, like TG is also very important biological parameter for everybody. Now, if the TG is high, then it also enhances the risk of stroke and heart attack, right. So, this just like for the testing. So, you can see like we have done very good study in terms of like at what pH value the enzyme is giving the maximum activities, right. So, then the pH value is 7.4. At that pH value black graph is there. So, you can see we are getting a maximum sensitivity of the sensor at the pH value of 7.4, right. And the best response sensor at 4 mg per ml solution of the lipase enzyme, like it is not like you have to use any concentration of the lipase enzyme, right. At this particularly concentration, you can get the better or maximum sensitivity, right. So, now I have spoken about the EDC NHS chemistry and covalent bonding, but one of the very good material, right, the scientists are using this MOS2 material in many like even in energy harvesting and in solar cell and in many other application people are using the MOS2. So, what we have done here, because if you are using this MOS2 and if you immobilize or coat on the optical fiber surface, so we do not need to have this EDC NHS chemistry, the covalent bonding is do not need, right, it is not needed. Directly the antibody is directly attached on the MOS2, that is the because of the surface, the free surface group, they are very good. So, basically you do not need any cross linker, right. So, this, so here you can see this molybdenum disulfide. So, people are using in many other application. So, we have prepared this nanosheet using the sonication techniques, right. So, using these nanosheets, we have used the MOS2 solution, and then they have attached on the SPR 5 sensor surface. So, you can see here. So, these are the like you can see we have used gold coated optical fiber, and then MOS2 is attached on this surface, because the gold is having very good affinity with the sulfur. So, then that is attaching very nicely, and then antibody of the bacteria is also immunocomplex, it is attached with the this MOS2 matrix. So, it is giving the two advantages. One is basically we are not applying any covalent bonding chemistry. So, directly it provide the attachment of the antibody with the free sulfur group, right, and it also enhancing the sensitivity, because the MOS2 is providing the bigger surface to volume ratio, right. so that having the more binding sites, right, more binding sites. So then that is giving the some of the good advantages. So these are the, like, we have first done the control experiments when there is no antibody, without antibody, like we have tested. So then you can also see there is no shift in wavelength, right, and though no significant change is evident, right. So here again we have done the comparative study, right. So you can see for the E. coli, like one is for the without antibody and one is for the with the antibody, right. So here you can see this is the lower graph, it is with the antibody and gold surface and fiber, right, and that is without the antibody. So these are the some of the more responses for the different bacteria, right. This is the typhimurium and that was the E. coli. So you can see their sensitivity is coming different for the different kind of antibody. Okay, and here we have compared using the anti-E. coli antibodies. So the first graph is basically for the E. coli detection, right, and these are the like 100 CFU and 1000 CFU and 2000 CFUs we have detected. This is again these are the selectivity basically. So you can see when we are using the this kind of sensors. So here the red graph is basically MOS2 antibody and the gold. So you can see the graph is going very you can see their response is very high. So then that is going straight, and then the black line is without the MOS2 or antibody. So from here you can conclude like we are getting the better sensitivity as well as the without the covalent bonding chemistry. So this is also it's a recent work, right, and then another kind of sensor, same MOS2 but for the detection of the protein, right. So we have detected the bovine protein, so using the anti-bovine antibody. So this we have published in the scientific report. So I hope we have some more time.
So what is the sensing length of this sensor?
Sensing length is around 2 centimeter.
Is there any variation if we will increase the sensing length or decrease the sensing length?
Actually only the thing is like sensing length you can increase or decrease, only the thing is like if you have a coating gold coating facility in such a way, so that if you can coat it, but it won't have any impact on the sensitivity, right. So sensitivity would be same, right, whether it is 1 centimeter or 2 centimeter, but in general because in SPR the sensing length is not much, right, because SPR babe is not traveling longer distances, right. It is die out in very short distance, right. So that is why the total length could be like your half a meter or something like that, the total length of your sensor, or maybe some 10-15 centimeter. So from the detector like spectrometer to the source, right. But the sensing where the gold is coated, and another thing is if you increase the sensing length, so you need a more antibody, and antibody is cost, yeah. No, there is no bending because it's a straight. This fiber is basically 600 micron fiber, right. It's not like 150 micron, right. It's a 600 micron fiber and 400 micron fiber. So that is what for the SPR people are using those fiber, right. So similarly, one can see from here these are the transmission spectrum, right, for the again. So now I come to this D fiber SPR biosensor. So here what we have done, we have edged the optical fiber. So D is basically, as you know, right, it can burn. So you have to use all the kind of protective gadgets, then only one has to use. So then, so we have used, these are the photograph of the D fiber, these are all the experimental results, right. So we have done all the experiments, right. And then the thing is, as compared to the normal SPR, like normal people are doing the SPR, this D fiber SPR is a more sensitive, right, because of this discontinuity in the edges, right. It basically it provide a better more enhancement of this more bi-components of the Poynting vector is crucial for the coupling of the plasmons, right. And if we do this kind of D, so they are coupling more efficiently, right. And these are the some of the coating and all those, so that I'm just going through just for the purpose of the their characterization, right. So here we have basically detected the CTC, they are the circulating tumor cells, right. So when there is some cancer or some tumor is starting the body, right. So at that release the circulating tumor cells, and they are traveling in the stream of your blood, right, and they can again go and they metastasis any part of the body. So the thing is like if it can be early detected, right, or maybe can be used for the screening purpose. So then people can know like yes, some kind of cells or tumor cells are there in the blood stream. So that can be, then it can be prevented. Like we have also then discussed with the oncologist in the PGI Chandigarh and all with our study, right. But the thing is their opinion is different because they are the doctors, so they told like we are doing the biopsy, and they are more comfortable the biopsy because under the microscopy they'll see whether the tumor is healthy or not healthy. So then, but of course we have the projects on this, so then we have demonstrated yes, this technique can detect, right. So then you can see like we have done this because this they are called the we have done HEP G2 cells, they are the basically liver cells, basically cancerous liver cells. So then those cells we have taken from Pune laboratory, and then we have used the antibody of this CTC. So only their concern is their main point is it going to be more costlier because of the you are using the antibody, and then it is also not that specific basically, right, because this antibody is basically attracting all other cells also, not only one particular, because there is a lot of different kind of cancers, right. So it can be a breast cancer or maybe some other kind of cancers, so they are not very specific, right. So aptamer technique is even more specific towards the to detect the cancer cells. So because they are aptamer people are developing for that particular kind of biomolecules, right. So you can see like this is we have tested and we have detected around 10 cells also and they are the all they are the spiked sample, right, like we have spiked them in the yes, human serum, and then that is how we have detected them. So the future is like as I have mentioned because I have also mentioned the futuristic like future perspective. So you see this sensor or this sensing platform, they can be more realistic and more useful if they can be integrated with the microfluidics, right. So using the microfluidic device, that you can, as you can see here, this is the glass substrate, gold film is made, and this is the micro channel. So from where your biomolecules is flowing, right, in a very small quantity and in a controlled environment. So then it can be made as a device kind of thing, so that it can be used for the such kind of purposes, right. So this is like one of the thing, like using the microfluidic chips with the surface plasmon resonance biosensor, it can be a more useful and more realistic. Okay. Photonics biosensor, if you talk of, so then now people are using the silicon photonics. Since silicon photonics, basically we are also working on the this micro ring resonators, right. So their dimension, it is looking a very big, but their dimension is of the order of the micrometer, right. This ring and all, so they are very, very good device, and then they can be made like this. So in one particular flow cell, the ring resonator is inside, and then one inlet and outlet, and then there is a detector. So this is a complete device, and then you have all those ring waveguide. So this is basically silicon photonics. For this, of course, this kind of to make or to fabricate such device, you need a very world class facility, that is called the clean room facility to fabricate such kind of things, right. Then nowadays you have also, if you have heard about the meta surfaces, so they are also engineering structures, special engineering structures which giving the special kind of functionalities, right. So they are called the this is our own work, but this is a simulating work, right, because we do not have the fabrication facility also in our laboratory. Now we are now developing. I'll show you in the next slide. So these kind of, and you can see their dimension is order of the 150 nanometer, 110 nanometer, and they are the basically asymmetric structures, and because of the asymmetricity, they are giving these kind of phonoresonances, right. And then if you basically work on this portion, right, here you can put your biomolecule. So then there is just change in this resonance wavelength, and then you can do the sensing. So the thing is, one ring is like this, and then the whole sensing platform is up to this, right. So these kind of, these are the futuristics. So then I thought of to show the some futuristic kind of research, so that people can also focus on such kind of research. So how to do the on chip photonics basically, right. So that in that direction we have started some of the work in our lab. So now we are establishing the clean room and the micro and nano fabrication facility in CSIO. It's one of the unique facility we are establishing in the North India. Like in any IIT or any other institution, no such facility is existing, right, because it is dedicated for the glass substrate, right, and silicon substrate, because people are having some limited, you can say, not bigger, but some limited kind of facility, but for the semiconductor, like
what class?
we are now we are class 1000 and class 10,000 both, right. So depending upon which substrate we are using, so then accordingly, because for the semiconductor chip we need to have a class 100 basically, right, and even if you go for the 30 nanometer technology, then people are using class 10, right. So but our purpose is to make the for the defense and strategic application, to make the beam combiner and to make the waveguides on the bigger substrate, right, about 6 inch and 10 inches, right. So that is very big platform. So such kind of things we are targeting. So as you can see, large area basically, right. So size is 10 nanometer to 10 micron feature size, right. So it can be like used for this such kind of application, like QPIC, quantum photonics integrated circuits, for the quantum technology. So basically, you know, like now our government of India and our honorable Prime Minister, they are very keen for this quantum technology, and they have also launched the National Quantum Mission. So many institutions, they are part of that quantum mission, and then we are also working on to develop the entangled photon source, right. So that to develop the entangled photon source or single photon source, you need to have such kind of facility to do the fabrication, okay. And then that can also use for the quantum logic gates and processor making, then CMOS integrated sensors, some of our colleagues, they are working on the 6G components using the terahertz devices, right? They are developed this terahertz detector and modulator, right? Our major focus is basically the laboratories has developed the head-up display for the Air Force, right? So that is our major this thing, right, footprint for the laboratory. And only one of the in India, only we are the people like in the India who's developed this for the fighter aircraft LCA, right? So now this is the conventional HUD, but using this such facility, now the target is to make the waveguide HUDs, then reduce the size and their weight and all those things, right? So this facility is very unique, which can cater the need of the many other institutions also, right? So using that facility, we are also targeting to make the micro lens arrays, micro nanophotonics components, meta surfaces, quantum optics, sensing. So these are the and you can see like so far even today, even in India, many our IIT like institutions through their startups, they are making spectrophotometer, right? So spectrophotometer, but still the components of the spectrophotometer is imported. The grating, which is a very essential part of the photometer, spectrophotometer, it is not made in India. So if such facility is there, then we can make those kind of gratings for the spectrometer, right? So that also like that also was one of the good step. You can say the way forward in some other to make the some other devices, right? So basic requirement is basically you have to build the clean room facility, micro and nano lithography tools. So we have EBL, it is already like in process to come, electron beam lithography. Then we have etching tool, so that the pattern can be transferred to the actual device. Then deposition tool, right, if you wanted to deposit some other material also, right, and then characterization tool. Of course, they are the very essential part of the process. So these are the application of the silicon photonics devices like lab on chip, biosensor, quantum computation, neural network, artificial intelligence, quantum key distribution, 5G and future network, and data center and communication. So these are the basically capability of the silicon photonics devices. These are the required simulation tools also, like the people, those who are working in the, like they don't have the experimental facility, so they can do some good simulation for the silicon photonics. So these are the very good platform like Photon Design, Synopsys, Numerical, OptiWave. So there are many tools, they are providing you a very good simulating platform, which can be later on realized in reality, right? So then that can be your idea and design can be tested, right? This is the one of the example I have taken from this thing, like this is very recent. It's asymmetric Mach Zehnder interferometer biosensor. It is already available online, right? So this is like it's a very most sensitive chip, right? Base transducers, and that detect the change in the refractive index or the order of the 10 to the power minus 8 refractive index unit, right? And it also overcome the inherent sensitivity limits in other sensing components like ring resonator with no theoretical limit to functionalize the sensor area available. So yeah, it has a very low noise and integrated signal reference, cancel out the noise from the environmental effects. So these are the like 8 arrays of the Mach Zehnder interferometer. So this is basically some photonic integrated circuits I have taken. I think it is in Canada or somewhere they have developed, right? So it's a very good sensing device, so which can go as a chip for the sensing biosensing application. So with this, thank you. I summarize, like I have demonstrated, I have just presented all this, right? And now acknowledge my director and some of my colleagues. With this, thank you. I don't took now I will not take much time because I'm in between you and your lunch.